We have to move to the next speaker, Dr. Claudio Fernandez, Max Planck Laboratory for Structural Biology, Germany. But I think that a particularly fruitful cooperation of a Max Planck partner group led to the establishment of a Max Planck lab in Rosario, Argentina, and Claudio is the director of this lab. Claudio is a biophysical chemist, specifically investigating the field of neurodegenerative diseases such as Alzheimer's and Parkinson's disease. The title of this talk is Reshaping the Role of Metal Ions on Alpha-Synuclein Aggregation. Please, Claudio, share your screen and start with your talk when you're ready. We cannot hear you, Claudio. Claudio, we cannot hear you. I don't know why.
Can you see now? Can you hear to me now?
Yeah.
Can you listen to me? Okay. Are you able to see the screen?
Yeah.
Okay.
You can start.
Okay, thank you. Thank you very much. I will then start with thank you, the organizer, for the invitation, and thank you for your nice introduction. I will talk about my focus of my talk will be on the role of metal ion on alpha-synuclein aggregation and the triggering of structural transition in the protein. So firstly, alpha-synuclein is an intrinsically disordered protein that it has 140 residues that are distributed in 30 well-defined regions. The alpha-hepatic region, that is the N-terminal binding domain, the NAC region, that is amyloidogenic itself, and this C-terminus, a region that is highly charged in negative residues, as aspartic and glutamic, for example, and it's involved in chaperone activities and also in rapid inhibition of amyloid assembly of alpha-synuclein. The protein exists in the cytoplasm of really in the presynaptic terminals in neurons in two forms: a free form that is intrinsically disordered, and it is adopts an ensemble of aggregation and inhibited conformations, and a membrane-bound form that is enriched in half a helical structure. And from these equilibriums depends the triggering of the aggregation intracellularly of alpha-synuclein. The physiological function of the protein is a matter of continuous debate. It seems that it should be related with the vesicle trafficking, with the release of neurotransmission, but it's still a matter of debate. Regarding to the links of the protein with the onset of the pathology, the protein is found to be the main component of Lewy body, that is the pathological hallmark of Parkinson's disease. In some years ago and continuously, it is happening also this year. There is point mutant that are familial mutants of alpha-synuclein that give rise to early onset of Parkinson's disease. And then when you make transgenic animal models overexpressing alpha-synuclein, this gives rise to cytoplasmatic inclusions that very similar to that found in the brain of post-mortem patients with Parkinson's disease. So in our laboratory, we studied the monomeric state of alpha-synuclein and the conformational change that triggers the transformation or the transition from this monomeric state to a species that is more prone to aggregate. As you can see here, as Parkinson's disease is a multi-factor disease, there's multiple factors that can trigger the disease. We studied all of these from in an interface, I would say in an interface that combine chemistry with biophysics and structural biology. So in that case, I will talk about the role of metal ions and why we started to study the role of metal ion. It is well now that metal ions can influence directly the structure of the protein or indirectly through oxidative damage, some of these metal ions, copper and zinc. But usually the role of metal ions are very related with the onset of neurodegenerative disorders, principally Alzheimer's disease. But when we started to work in Parkinson's disease and with alpha-synuclein, this was a complete vacant field we had to develop. So we use a combined strategy that mainly uses NMR spectroscopy. This is a two-dimensional map in which each cross peak that you can see here represent to an amino acid, and a specific amino acid in the protein primary sequence. So the starting point for this was, okay, this is an intrinsically disordered protein. This protein lacks rich secondary structure. This protein lacks tertiary structure. So which is the starting point? So we use some paramagnetic stacks that derives from nitroxide spin level, and we located this paramagnetic nitroxide at different parts of the protein. The sphere of action of this nitroxide was around 25 angstrom from their location. So from this, we built interaction profiles, and from these interaction profiles, we knew and we learned that the monomeric state of alpha-synuclein was stabilized by long interaction that were of transient nature between the C-terminus and N-terminus that were of electrostatic nature, and between the C-terminus and the NAC region that were of hydrophobic nature. And then if you destabilize this transient long-range interaction, you give rise to a population of an aggregation-prone state, and the aggregation is trivial. And the most important is this scheme that I want to share with you, that in which shows that the C-terminus of the protein is folding back over the NAC region, over the central region, and protect the central region to be exposed and to trigger aggregation. This is the native conformation of alpha-synuclein in the cytosolic free state. So what happened with the metal ions? When we started to study these metal ions, we found a lot of published articles that related to that a lot of divalent metal ions were able to induce aggregation, to accelerate the aggregation of the protein, but using millimolar concentration of metal ions. So we tried to make a differentiation with all these metal ions. And in this study, we use micromolar concentration of these metal ions. So under this micromolar concentration of metal ions, copper in the oxidative state, copper 2 was the only divalent metal ion that was able to accelerate the aggregation of the protein from here to here. The other divalent metal ions were not able to accelerate this the aggregation of the protein under these experimental conditions. So we wonder why, and so we learned that we studied the interaction between this protein and different metal ions, manganese, cobalt, all of these calcium, cobalt and manganese were very useful because they are paramagnetic metal ion. So in the presence of this metal ion, due to the paramagnetism of the metal ion, the signal disappears, then you can map the interaction. So what we learned that these metal ions in the millimolar levels were able to bind exclusively to the C-terminal region of the protein by an specific electrostatic interaction. And what happened when we analyzed this interaction profile is that upon binding of these metal ions to the C-terminal region, there is a release of this C-terminal region from the native state. So this is not folded back anymore. The NAC region, the hydrophobic region, is exposed to the solvent, and there is an opening of the transient structure of alpha-synuclein. And then there is an interaction between the different monomers, and aggregation is triggered. So this is a general mechanism by which some metal ions, mainly divalent metal ions, induce aggregation and accelerate the aggregation of the protein in the millimolar range. But we wonder why the divalent metal ion, copper-2, were able to induce aggregation of alpha-synuclein in a micromolar range. Indeed, we use 10 to 30 micromolar concentration of copper level that were able to accelerate the aggregation of the protein. So we have here mapped the situation with the divalent metal ions. Most of these metal ions are able to bind to alpha-synuclein with interaction in the millimolar range. So you need millimolar concentration of metal ions to accelerate the aggregation of alpha-synuclein. In the case of copper, you can, of course, trigger the aggregation using millimolar concentration of the metal ion, because this metal ion is also able to interact with the DC terminus region. But added to that, in addition to this, there are two copper binding sites in the N-terminal region of the protein, involving the N-terminus and methionine and aspartic position 2, and the other one involving histidine residue. So this is the picture of the interaction profile of copper with copper-2 with alpha-synuclein. And at this stage, we wonder, okay, which would be the affinity, the molecular mechanism behind the acceleration of alpha-synuclein aggregation by copper? Of course, we think immediately in this high affinity binding site, but maybe also this binding site involving the histidine residue could be involved. So we prepare a series of mutants which were C-terminal truncated, so we don't have the specific binding site at the C-terminus. We also lack the histidine binding site. And so under this condition, it was we could demonstrate conclusively that it is the binding of copper to the high affinity binding sites at the N-terminus of alpha-synuclein, which is a factor that triggers the aggregation of the protein. So the aggregation, the acceleration of the aggregation of alpha-synuclein is mediated by copper binding to the high affinity binding site in the N-terminus. That involves coordination to the amine nitrogen of methionine at position now, an oxygen coming from a water molecule, an oxygen coming from the side chain of aspartic at position 2, and the amine atom of aspartic at position 2. Of course, we had to develop a strategy in which combined nuclear magnetic resonance with molecular dynamics, with Malditov, with electronic paramagnetic resonance. So this is the picture that we know, and why the effect of copper at the low micromolar concentrations is due to the fact that the copper is able to interact more specifically with alpha-synuclein than the other studied divalent metal ions. So next step was, okay, which would be the mechanism? Could be mediated by an oxidative damage, protein aggregation, and neuronal cell death. That is one of the potential mechanisms by which metal ions are involved in neurodegeneration, or could be a consequence of a direct interaction between copper and the protein, and a transition that is triggered because of the interaction towards a structure that is more prone to aggregate. If you observe here the 2D map, two-dimensional HSQC experiment, in which each cross peak represents an amide group in the backbone of alpha-synuclein, in the presence of copper, there is a disappearance of some spectra because of the broadening induced due to the coordination of copper to that binding site. But this is not, there is not a chemical shift, a dramatic change in chemical shift. So it is a proof, it is an evidence that there is at least not an evident transition or structural transition driven by the interaction between copper and alpha-synuclein. So we try to explore, to assess the the effect of oxidative damage induced by copper, but for what we had to work with copper 1, and we had to show that the protein were able to bind also the reduced state of copper. And so we performed a very, very easy experiment without any need to label the protein for doing NMR experiment in nitrogen-enriched alpha-synuclein. This is 1D experiment. This signal corresponds to the 4th methionines in alpha-synuclein, and you see that in the presence of copper, there is a reduction and a loss of intensity of 2 of these histidines, which means that copper was able to interact with these 2 methionines, with these 2 methionines at position 1 and position 5, and after air exposure, probably induced the oxidation damage. We then monitor this and at different time intervals in which we show that the protein were indeed oxidized, both methionine at position 1 and 5 to methionine sulfoxides in the presence of copper complexes and high concentration of a reductant agent that we use ascorbic acid, and exposure to air to the different time intervals. So we also show this by using multi-mass spectrometry, and we conclude that indeed there was an oxidative damage induced by the copper 1, copper 2 redox couple, that both metal ions, both states of the metal ions were able to interact with the protein. And of course, we also show that that the protein, the oxidative damage of the protein was able to impair the ability of the protein to interact with the membrane, and it also give rise to impairment of amyloid fiber formation. And instead of that, the protein was going also to the formation of oligomers that were extremely toxic. After that, we decided to explore in detail the interaction between copper 1 and alpha-synuclein, because physiologically the copper 1 interaction is the most relevant one. So we combined this study with different point mutants of alpha-synuclein. We also find 3 binding sites in the N-terminal region and in the C-terminal region that involve mainly methionines and histidines. This is just to show that how useful are the combination of NMR study using point mutants of the involving residue in copper coordination. We show by this study that the high affinity binding site for copper 1 in alpha-synuclein involve the sulfur atoms of methionine at position 1 and 5. We then combine this study with different peptides in which the methionine were replaced by ileosin, and the replacement of both methionine give rise to the lack of interaction of copper with the peptide. So it means that both methionine at position 1 and 5 are key for the binding of copper 1 to the protein. Having arrived at this, we decided to study also which was the impact of some post-translational modification on the metal binding capability of alpha-synuclein. And the reason why we studied that is because we found doing in cellular NMR that the protein was mostly acetylated in the N-terminus. So which was the impact of this acetylation in interaction with copper 2? Okay, upon acetylation of the protein, we observe that there is a lack of the interaction of the high affinity binding sites. In the case of copper, it was amazing because we observed in the case of copper 1 that the affinity for the high affinity site was increased by a factor of 2, but also was something very, very atypical, very important, that is a dramatic change in the conformation of the protein trigger upon binding of copper-1 to the high affinity binding site by using some specific NMR parameters that are related to structure, that is the chemical shift of alpha carbons or beta carbons, or the chain couple between alpha protons and amine proton. We found that there was an induction and a stabilization of an alpha helical segment in alpha-synuclein extending from residue 1 to 10 upon binding of copper. When we combine this with relaxation measurements, we give you idea of the rotational dynamics. We observed that there was an increase in the rigidity of this region. It means there is a loss of flexibility that was related with the stabilization of the alpha helical conformation in that site by the binding of copper, that was triggered by the binding of copper. And this is what happened with the protein upon binding of copper. And in addition, we found that there is an equilibrium between two forms of the protein in which there is a transient dynamically alpha helical conformation that extended from residue 1 to 50 to 25 in the presence of copper-1. When we study the role of the different residues, we found that methionine, by combining these studies with different mutants, we see that when this double mutant, there is a lack of interaction here. There is not change the position of the peaks mainly, but here remains the interaction. So we, from the combination of these interaction profiles by NMR, and in combination with relaxation data, you see that in the case of this mutant, there is no such a rigidity here. There is the loss of flexibility is less pronounced in this case. And it means that, but there is interaction. So it means that methionine at position 1 and position 5 are key factors for determining the affinity of binding of copper-1 to alpha-synuclein, whereas the acetylation and the aspartic at position 2 are key for stabilization of the alpha helical conformation triggered by copper-1, sorry. Added to that, I had to say that this stabilization, this triggering, this transitional change that occurs after copper binding give and only occurs in the acetylated form of alpha-synuclein. It does not occur, it does not take place when the protein is not acetylated at the end terminus. And finally, we study the effect of the interaction of copper-1 with beta-synuclein. And the reason why we study beta-synuclein, it is because beta-synuclein is a member of the family of alpha-synuclein. This is the only member that is co-localized with alpha-synuclein in the neurons, and it has neuroprotective effects. However, this was not detected any kind of interaction, direct interaction between beta-synuclein and alpha-synuclein. So we wonder if the affinity of the copper by beta-synuclein could be higher, if this protein could act as a sequestrating copper and protecting copper for interacting with alpha-synuclein. And interestingly, you mean that compare with alpha-synuclein at the end terminus, that both proteins have a high homology at this N-terminal region, there is an extra methionine at position 10 in beta-synuclein. We studied the interaction with copper-1. This is the interaction profile, and we found that the 3 methionine at position 1, 5, and 10 were involved in copper binding, and the copper binding was higher, 10 times higher than the high affinity binding site in alpha-synuclein. So we then explore if this interaction could be raised to a conformational transition as the one found in alpha-synuclein. To our surprise, we found that there is not any important relevant transition in a case of beta-synuclein, that in this case, the only change was restricted to the first 5 amino acids in the sequence. And the reason why is because due to the complexation of the extra methionine at position 10, there occurs a bend, a turn in the protein around glycine at position 7, and this bend, this turn around glycine at position 7 give rise to a break in the alpha helix. So it is not possible in beta-synuclein to induce a conformational transition of the magnitude of the one observed in alpha-synuclein. And the next step that we are exploring now is what happened in cellular models that are positive for inclusions, intracellular inclusion of alpha-synuclein, combining copper with beta and alpha-synuclein that has been and are being transfected into these cell models. And I want to thank my collaborators in Germany, Thiago Oteiro, Marcos Ostetter, Christian Grisinger, and also in Germany, Paolo Carloni, Liliana Quintanar in Mexico, also Nicolas Rey in Brazil. In combination with Nicolas Rey, we are developing some metal attenuating compounds. Some works have been published already, and some works are currently be performing. Thank you very much for the attention.
Thank you very much, Claudio, for your really inspiring talk. And now we have time for some questions from the floor. Okay, there is a question that's sorry from an anonymous attendee, but sorry, Claudio, but synuclein doesn't look like that with a hole in the middle. I really don't know what structure this anonymous attendee is talking about. However, Claudio, if you want to try to answer to this remark.
The structure of alpha-synuclein has been the matter of debate. I mean, when we determine the structural feature of alpha-synuclein, we have to be very careful regarding to that because it's an intrinsically disordered protein. So we talk about the propensity to adopt some structure. We are not talking about rigid structure. We found that the protein adopted an ensemble of auto-inhibited conformation in which the C-terminus region of alpha-synuclein were interacting with the N-terminus region of alpha-synuclein, and this interaction was mainly based on electrostatic interaction. The other long-range interaction stabilizing this conformation was between the NAC region and the C-terminus region, and was of hydrophobic nature. By using in-cell NMR some years after, it was shown that the protein adopted exactly the same structure intracellularly. Even if there was some conclusions, some works reporting the existence of a tetrameric form of alpha-synuclein, several groups of research, the main groups of research working in the structural biology and biophysics of alpha-synuclein were not able, and I include my group, to detect the tetrameric form of alpha-synuclein. And in addition to that, the protein exists also in a membrane-bound state. And between the equilibrium between the membrane-bound state conformation and the cytosolic free conformation of the protein, this equilibrium is determined in the aggregation capability of the protein intracellularly. This is commonly accepted now. And regarding to the conformation of the protein bound to the membrane, we can also discuss about this. I mean, it depends on the composition of the phospholipid. It depends on the curvature of the lipid membrane. It depends also of a lot of parameters. this is now accepted that those states in which the protein bound to the membrane, in which the NAC region is detached, is released from the membrane-bound surface, is more amyloidogenic and more toxic, and has more capability to aggregate that the bound state in which all the first 100 residues are involved in the interaction with the membrane surface.
Okay, thank you, Claudio, for your very exhaustive answer. And I think that we are perfectly on time, and we can go to the 10-minute break, where we can, if possible, we can retrieve some left out questions from the first or second speakers. I think that there are 2 left out questions, but I don't know whether Astrid is still in the panel, because I think that these 2 left out questions were for Astrid. No, I don't see Astrid, so I think that we can, okay, we can. No, we have another, sorry, okay. We have some questions from Rams. Very nice talk, Claudio. Is the helical structure 125 residues of alpha-synuclein that showed experimentally solved? Is it by NMR? And the second question is, how does copper binding to residues 1,5 get the 125 to become helical?
This is a good question, very nice question. I mean, yes, the structure of the membrane-bound state of alpha-synuclein is already solved and is determined by NMR spectroscopy. Yes. Regarding to the interaction of copper with the membrane-bound state of alpha-synuclein, I guess that the question is copper 2 or copper 1. I don't know. I mean, I really don't know. Copper 2 mainly interacts with the C-terminal region, because the C-terminal region and the membrane-bound state is not interactive with the membrane phase, with the membrane surface. So it's free, so it can interact with copper 2. It's very difficult. It's very difficult to see to study this interaction by NMR spectroscopy, because mainly there is a weakening of the intensity of the first 100 amino acids upon interaction with the membrane. Fortunately, I cannot talk too much about the interaction with these 2 metal ions in the membrane-bound state of the protein, because it is unpublished results.
Okay, thank you. And we have another question from Sanil Kumar. Fantastic work. I wonder if you could talk a little about the affinity of synuclein with copper in the absence and presence of lipid membranes, and if the bind of copper to synuclein regulate the function of membrane-bound synuclein.
Well, I will say, because I think that we are here to exchange and to communicate science. So I can tell you. I mean, yes, there is a higher affinity this is high affinity interaction is highest that the protein in the free in the unbound state, and it is because the triggering of a helical change in alpha-synuclein. So you have a copper bound to alpha-synuclein, copper 1 induce and stabilize alpha-helical conformation, and so these are of higher affinity for the membranes. And yes, I think that we found evidences that the copper interaction with alpha-synuclein regulates the structure of the bound state of alpha-synuclein, and of course, if it should also regulate the function.